Hello, and thank you for attending our presentation. I am Gary Bolin, a professor of biomedical engineering at the University of Memphis. Today's talk by our laboratory will be presented by my PhD student, Ms. Allison Fetz, who conducted the work being presented. We would like to thank the organizers for the opportunity to participate in this session. Hope all will remain well and enjoy the session. Thank you.
Hello, everyone. Before I get into my talk, I want to say thank you to Dr. Bolin for giving me the chance to present at this meeting. And as Dr. Bolin said, thank you to the meeting organizers for coming up with a way to bring us researchers together to discuss our work while remaining safe and healthy during this pandemic. Today, I'm going to talk to you about some of our recent work with Electrospun biomaterial drug delivery systems, titled Electrospun Polydioxanone Biomaterials Loaded with Chloroquine, Modulate Biomaterial. Hello, everyone. Before I get into my talk, I want to say thank you to Dr. Bolin for giving me the chance to present at this meeting, and thank you to the meeting organizers for coming up with a way to bring researchers together to discuss our work while remaining safe and healthy during this pandemic. Today, I'm going to talk to you about some of our recent work with Electrospun biomaterial drug delivery systems, titled Electrospun Polydioxanone Templates Loaded with Chloroquine, Modulate Template-Induced Net Release, and the Inflammatory Response. To begin, our lab is largely a tissue engineering lab, so we approach biomaterial design with the end goal of creating a biomaterial that will stimulate and guide functional tissue regeneration. However, the greatest challenge for the clinical translation of biomaterials and tissue regeneration applications is guiding the integrative and functional tissue regeneration we desire because of the biological response to these biomaterials. In recent years, we've been focused on the early stage events and the biological response. So let's take a closer look at what happens when a biomaterial is implanted. During the biological response, proteins absorb on the surface of a biomaterial immediately following its implantation through a process known as the Roman effect. Following protein absorption, immune cells then interact with the surface absorbed proteins and respond. This is known as outside-in receptor signaling, which transduces microenvironmental cues into intracellular signals and a subsequent cellular response, effectively linking the cell response to the properties of the biomaterial. The summation of these immune cell responses then regulates the extent of the foreign body reaction and the potential for fibrotic tissue development or tissue integration and regeneration. One important aspect of this foreign body reaction is acute inflammation, which occurs very rapidly and is characterized by an influx of neutrophils to the environment around a biomaterial. Because of the neutrophils' role in acute inflammation and our interest in tissue regeneration, our group has spent the last several years focusing on neutrophil biomaterial interactions. Neutrophils are the most abundant white blood cell on the body that interact first with the biomaterial. While it is accepted that the outcome after implantation of a biomaterial is largely dependent on the extent of the foreign body reaction, it is becoming increasingly recognized that the neutrophil plays a central role in this outcome. Classical responses that shape the biomaterial environment include phagocytosis, degranulation, and the generation of reactive oxygen species, which together modulate acute inflammation and the extent of the foreign body reaction. In addition to these well-described cellular responses, neutrophils can also release neutrophil extracellular traps, or nets. Nets are three-dimensional structures composed of decondensed chromatin, histones, proteases, and other antimicrobial factors. They're released through a specialized form of cell death called netosis for the purpose of trapping and killing invading pathogens. While nets are beneficial for combating infection, the dysregulated release of nets can be detrimental and is associated with chronic inflammation, autoimmune disorders, tissue fibrosis, and thrombosis, suggesting that in the context of biomaterials and tissue regeneration, nets may be harmful. In fact, nets are also released in response to biomaterials, including titanium plates, nanoparticles, and electrospun biomaterials. Using human neutrophils, our group has recently shown that nets are differentially released on the surface of electrospun polydioxanone biomaterials based on their topographical cues. In particular, we found that small electrospun fibers increase the propensity of neutrophils to form nets, as shown here with nets stained green in the top right fluorescent micrograph, whereas large electrospun fibers decrease the propensity of neutrophils to form nets, which can be seen in the lower right-hand micrograph with more intact nuclei. Moreover, we found that the increased presence of nets on small fiber electrospun biomaterials impairs tissue integration and promotes fibrotic tissue development in a rat subcutaneous implant model. This can be seen in the histology sections below, where the biomaterial is marked with an X. It's surrounded by a pink fibrotic capsule in the top image, but is integrated with surrounding tissue in the bottom image. Given our end goal of creating a biomaterial that will stimulate and guide functional tissue regeneration, our data indicates the upregulated release of nets will promote fibrosis. So the goal of this work was to develop a drug delivery system to inhibit net release as part of acute inflammation in order to increase the potential for tissue healing. The drug we selected for this work as our biomaterial additive is chloroquine. Chloroquine is an FDA-approved anti-malarial drug that has more recently been investigated as an immunomodulatory and antithrombotic drug. Specifically, it has been used to treat systemic inflammatory conditions such as rheumatoid arthritis, systemic lupus erythematosus, and cancer. Recently, chloroquine has also been shown to inhibit net formation, indicating its potential benefit as an additive for regulating biomaterial-induced net release and inflammation. In this work, we developed chloroquine-loaded electrospun PDO biomaterials and evaluated their ability to modulate acute human neutrophil inflammatory responses in vitro. We first studied the ability of chloroquine-eluting biomaterials to inhibit net release. Then, because of clinical evidence highlighting chloroquine's immunomodulatory effects, we also evaluated the ability of chloroquine-eluting biomaterials to regulate the secretion of pro-inflammatory and pro-healing mediators from human neutrophils. Our first task in this work was to develop chloroquine-eluting biomaterials. Because of our prior studies demonstrating that small and large electrospun fibers differentially regulate net formation, we began by formulating solutions to create both small and large fiber biomaterials as controls. Then we took these solutions and added chloroquine at a concentration of 0.07 milligrams per mil, so we ended up with 4 unique solutions. These solutions were then electrospun with optimized parameters, and the resulting biomaterials were characterized for fiber size by scanning electron microscopy and chloroquine elution through an absorbance-based assay. Let's take a closer look at these biomaterials. For all 4 of our biomaterials, the electrospun fibers were uniform, and we controlled the fiber size during electrospinning so that they did not change with the incorporation of chloroquine. This is important since the topographical cues of electrospun fibers modulate net formation, so any changes we see here can be attributed to the elution of chloroquine. Now, looking at our results from the elution studies, both of the chloroquine-loaded biomaterials eluted nearly 100% of the drug within the first hour. This solution equated to concentrations near 12 micromolar for both the small and large fiber biomaterials. It's important to note that we optimized these elution profiles to be rapid and to approximate previously reported concentrations shown to inhibit net release. As the focus of our work was on regulating acute neutrophil biomaterial interactions, this rapid delivery to therapeutic concentration was exactly what we wanted. Now that we had our biomaterials developed to deliver chloroquine, we studied them in vitro with human neutrophils. Peripheral blood neutrophils were isolated from the whole blood of healthy donors by density centrifugation. The isolated neutrophils were then cultured on the biomaterials for 3 and 6 hours under standard culture conditions. Subsequently, we quantified the release of nets with a net-based ELISA and quantified the secretion of a panel of inflammatory and regenerative mediators by an immunomagnetic assay. Now let's take a closer look at some of our results. As anticipated, neutrophils had an increased propensity to form nets on the small fibers compared to the large fibers at 3 hours. This can be seen in the fluorescent micrographs where the nets here are shown red and the nuclei are shown purple. More importantly, the elution of chloroquine from the small fiber biomaterials significantly reduced that release to the level of the large fibers at 3 hours, while having no effect on the large fibers. Although we cannot explain this difference in efficacy, we observed similar results in our prior work using another drug to inhibit net release, suggesting that some other mechanism of regulation is involved in these responses. In addition, by 6 hours, the difference in net release was less pronounced, indicating a temporal therapeutic window for inhibiting acute net release. Taken together, these data illustrate chloroquine elution from an electrospun biomaterial does in fact inhibit net release, specifically on our small fiber biomaterials. In addition to looking at net release, we examined the potential of chloroquine to modulate soluble signal secretion from neutrophils. First, I'm going to show you data for inflammatory signals before we look at some of our data for regenerative signals. The 2 inflammatory signals modulated by chloroquine were interleukin-8, or IL-8, and matrix metalloproteinase 9, or MMP9. At both 3 and 6 hours, IL-8 secretion was significantly greater on the small fiber biomaterials compared to the large fibers. This mimics the same trends as net release, which really is not of great surprise since both can be thought of as inflammatory responses. Furthermore, the elution of chloroquine significantly suppresses IL-8 secretion. Since IL-8 is the archetypal neutrophil chemotactic, this may be important for regulating aberrant neutrophil chemotaxis during the acute inflammatory response. As for MMP9, similar results were observed, but the level of secretion was near equivalent on all biomaterials by 6 hours, suggesting a temporal modulation of MMP9. Nonetheless, because MMP9 is involved in extracellular matrix degradation, its suppression by chloroquine could help prevent acute excessive matrix destruction that perpetuates chronic inflammation. We also looked at how chloroquine-eluting biomaterials regulated regenerative signal secretion. Chloroquine is not well studied for its potential regenerative effects, but we were interested if its immunomodulatory effects extend beyond its suppression of inflammation. In these experiments, we found that the secretion of 3 factors were modulated by chloroquine: hepatocyte growth factor, or HGF, vascular endothelial growth factor A, or VEGF, and interleukin-22, or IL-22. The secretion of HGF and VEGF followed very similar trends, with both having significantly greater secretion on large fibers compared to small fibers. This is the inverse trend of net release and pro-inflammatory factor secretion, indicating that the large fiber biomaterials alone may promote more of a regenerative neutrophil phenotype. Additionally, chloroquine elution from both the small and large fiber biomaterials increased the secretion of HGF and VEGF to the level of the large fibers. HGF and VEGF are both pro-angiogenic factors, so their increased secretion could aid in resolution and tissue regeneration during the acute inflammatory response. Likewise, IL-22 followed similar trends, but was not detected until 6 hours, probably due to an absence of readily available stores. IL-22 is also a pro-angiogenic signal, so together the modulation of HGF, VEGF, and IL-22 by chloroquine suggests a previously unrecognized aspect of chloroquine's immunomodulatory effects by increasing regenerative signal secretion. To quickly recap, the primary goal of our lab is to develop biomaterials that stimulate and guide functional tissue regeneration. But the greatest challenge for the clinical translation of these biomaterials in tissue regeneration applications is guiding functional tissue regeneration. Nonetheless, Electrospun biomaterials are excellent candidates for drug delivery systems to modulate acute inflammation and increase the potential for tissue regeneration. In recent years, our group has been largely focused on regulating the neutrophil response to biomaterials, showing that Electrospun fiber size regulates the release of nets and that increased nets promotes tissue fibrosis. In this work, we showed that chloroquine can be used as a template additive to modulate acute neutrophil biomaterial interactions. More specifically, we showed that chloroquine elution decreases biomaterial-induced net release on small fibers and decreases pro-inflammatory IL-8 and MMP9 secretion, regardless of fiber size. At the same time, we also showed that chloroquine elution increases the secretion of pro-healing factors HGF, VEGF, and IL-22. Taken together, these data illustrate the potential therapeutic benefit of chloroquine in Electrospun biomaterial applications. Because this work was performed in vitro, there is still work to be done. Our data indicate that the local delivery of chloroquine through Electrospun biomaterials may downregulate acute neutrophil-driven inflammation, while simultaneously upregulating the neutrophil regenerative phenotype. Next, it will be necessary to evaluate the in vivo efficacy of our chloroquine-eluting biomaterials and study the mechanisms of regulation involved. Nonetheless, through our in vitro studies, we have shown that our chloroquine-eluting biomaterials regulate acute neutrophil-driven inflammatory responses by downregulating net release and inflammatory signals while upregulating regenerative signals. And ultimately, these responses may have synergistic effects that are advantageous for diverse Electrospun biomaterial applications. With that, I would like to acknowledge my lab, in particular my advisor, Dr. Bullen, my former lab mate, Ben Minden-Birkenmeyer, and Hunter King and Shannon Wallace for their help on this project, as well as the Radic Lab, who are the experts in neutrophil biology and collaborators who have guided us through our work with neutrophils. Thank you for your attention, and I look forward to answering any questions you may have.